This video is part 2 of Chapter 9, the Muscular System. In the first video, we learned about the different types of muscle tissue. In this video, we will focus strictly on skeletal muscle. Specifically, we will learn about its anatomy. We will describe the connective tissue components of skeletal muscle, and we will dive into the components of a muscle fiber, including the myofilament arrangement in the sarcomere. Let's begin with the connective tissue of skeletal muscle. Remember that an organ consists of 2 or more tissue types working together for a common function. A skeletal muscle is made of several types of tissue, not just muscle tissue. A skeletal muscle consists of skeletal muscle tissue, nervous tissue, and connective tissue. We are starting our discussion by examining how connective tissues play a role in skeletal muscle anatomy. Covering the outside of the whole skeletal muscle is a connective tissue sheath called the epimysium. The epimysium's fibers merge with the muscular fascia, which is the connective tissue layer between adjacent muscles or between muscles and skin. The perimysium is connective tissue that divides the whole muscle into bundles of muscle cells. These bundles of muscle cells are called fascicles. In this image, the perimysium is the light pink structure. The last connective tissue covering is the endomysium, which is the delicate layer that surrounds individual muscle fibers in a fascicle. In this image, it is the purple ring at the end of the arrow. Nervous tissue is the other type of tissue in a skeletal muscle. Skeletal muscles contract in response to motor neurons. Motor neurons originate in the brain and spinal cord. Each muscle is supplied by several motor neurons. One motor neuron branches to innervate several muscle fibers or cells. Each cell is innervated by its own branch. Next, we will dive deep into the skeletal muscle to learn about muscle fibers or muscle cell anatomy. Muscular cells are also known as muscle fibers. They contain numerous mitochondria and glycogen, the glucose storage molecule. Having both numerous mitochondria and glycogen molecules allows muscle cells to continuously produce enough ATP for the muscle to contract. Additionally, the cytoplasm of muscle cells is called sarcoplasm. A muscle contraction has both an electrical and a mechanical component. So the remainder of the structures in a muscle fiber will be categorized into the role they play in a muscle contraction. We will first learn the electrical structures. Electrical structures are the muscle fiber components that respond to and transmit electrical signals. Here we see a diagram of one muscle cell, or you can call it a muscle fiber. The plasma membrane of a muscle fiber is called the sarcolemma. Electrical signals are transmitted along the sarcolemma. In the image, the sarcolemma is salmon colored and is covering the entire cell. At some places along the muscle cell, the sarcolemma folds inward towards the center of the cell. These are called transverse or T tubules. In the image, the T-tubule is a lime green color. T-tubules carry the electrical impulse from the sarcolemma into the cell's center. The sarcoplasmic reticulum, shown in orange in the image, is a specialized smooth endoplasmic reticulum that stores calcium necessary for muscle contraction. The area of the sarcoplasmic reticulum that is next to a T-tubule is called a terminal cisterna. A triad is two terminal cisternae and the T-tubule located between them. Mechanical structures are the muscle fiber components that allow muscles to contract. As a reminder, this image is of one muscle fiber. Myofibrils are bundles of protein filaments. Each muscle cell has numerous myofibrils in its sarcoplasm. They run the length of the muscle cell. In this image, the cell has 4 myofibrils. The top image shows a muscle cell with 5 myofibrils. The bottom image is a close-up of 1 myofibril. Myofibrils contain smaller structures called myofilaments. 2 types of myofilaments are found in a myofibril. Shown at the bottom of this image, actin is the thin myofilament, while myosin is the thick filament. In our discussion, actin is shown in purple and myosin in green. This image shows a close-up of a myofibril. In the myofibril, the myofilaments are arranged in a pattern that results in what's called a sarcomere. Sarcomeres are the smallest portion of a muscle that can contract. This makes sarcomeres the functional unit of muscle fiber. Z-discs are stationary zigzagging proteins that actin attaches to. The top image shows the zigzagging Z-disc at the yellow arrow. Sarcomeres extend from one Z-disc to the next Z-disc. The red arrow indicates where the sarcomere is found in the myofibril. During contraction, the sarcomere shortens. M lines are the proteins that myosin attaches to. The blue arrow in the bottom image shows where the M line is located. You can see the green-colored myosin attached to the M line. Remember the striations or stripes we saw when we looked at a skeletal muscle slide? The striated appearance of skeletal muscle is due to the arrangement of actin and myosin in the myofibrils. The lighter areas are called I bands. This region contains a Z-disc and only actin myofilaments. A bands are the central part of the sarcomere that is a dark striation. A bands contain the overlapping actin and myosin myofilaments. Titin is a long, thin protein that runs from an M line to a Z-disc. At the M line, titin runs below it and anchors myosin to it, keeping myosin centered in the myofibril. Titin also anchors myosin at the Z-disc, but at the Z-disc, titin is shaped like a spring. This allows sarcomeres to recoil back to their resting length after being stretched. For this reason, titin is the main reason muscles have extensibility and elasticity. Let's learn a little bit more about the myofilaments. Actin is the thin myofilament shown in purple in the top image. It is made up of 3 separate protein components, and those can best be seen in the lower images. Globular actin, which look like purple balls in the image, have active sites. Tropomyosin is shown in white in the middle image and covers the active sites when the skeletal muscle is relaxed. When tropomyosin is covering the active sites, muscle contraction cannot happen. Lastly, troponin, shown in red, has a calcium binding site and controls whether tropomyosin is covering active sites or not. Myosin myofilaments are the thick filaments and are shown in green in this image. These elongated structures have many golf club shaped heads. The heads are hinged so they can pivot the direction that they face. When tropomyosin is not covering the active sites, myosin heads attach to the active sites on actin to form cross bridges that work to contract the muscle. Myosin heads also attach and hydrolyze ATP to release the energy needed for muscle contraction to occur. In this video, we learned the connective tissue components of skeletal muscle. We also learned the components of a muscle fiber, including the myofilament arrangement in a sarcomere.